Hello everyone, I'm Dr. Varuni Koskallana here to talk to you about the molecular basis of cancer or carcinogenesis. Non-lethal genetic damage lies in the heart of carcinogenesis that leads to abnormal cells with abnormal proliferation. These may occur due to carcinogens such as chemicals, radiation, or microbial agents. There are four classes of normal regulatory genes that are the principal targets of these carcinogens. They are the growth-promoting proto-oncogenes, growth-inhibiting tumor suppressor genes, genes that regulate programmed cell death or apoptosis, and genes involved in DNA repair. Proto-oncogenes present in normal cells are required for normal cell growth and normal cell differentiation. Mutation of these will lead to oncogenes, resulting in abnormal cell growth and abnormal cell differentiation. Then we have the tumor suppressor genes. Cells divide by going through the cell cycle, the phases of gap1, synthesis, gap2, and mitosis, resulting in identical daughter cells. In normal cells, tumor suppressor genes control cell growth and division by regulating the cell cycle, entering the resting stage, undergoing DNA repair, or resulting in apoptosis. Mutation of these tumor suppressor genes will cause dysregulation of the cell cycle, resulting in uncontrolled cell growth and division. Now about the genes that regulate apoptosis. In normal cells, if DNA damage occurs, genes that regulate apoptosis will cause programmed cell death. When there is a mutation of these genes that regulate apoptosis, with DNA damage, there will be accumulation of mutations and uncontrolled cell growth and division will result. Last but not least are the genes involved in DNA repair. In normal cells, when there is DNA damage, the DNA repair genes will act to correct and repair the DNA damage. In case of mutation, there will be no DNA repair, leading to accumulation of mutations. Thus, uncontrolled abnormal cell growth and division will occur. In summary, we learned about the four classes of normal regulatory genes which are the principal targets of carcinogens. Growth-promoting proto-oncogenes, growth-inhibiting tumor suppressor genes, genes that regulate programmed cell death or apoptosis, genes involved in DNA repair. Thank you.